Telomerase lipid nanoparticles. Background: telomerase and lipid nanoparticle therapy. Telomerase is an enzyme composed of the catalytic protein TERT and an RNA template that adds DNA repeats to telomeres, the protective caps on chromosome ends. In most human somatic cells, telomerase is inactive, so telomeres shorten with each cell division, eventually leading to cell senescence or death. Telomere shortening is a key mechanism of cellular aging and is implicated in age-related diseases. The idea behind telomerase therapy is to transiently reactivate TERT in cells to lengthen telomeres, thereby reversing or delaying cellular senescence without permanently altering the genome. Lipid nanoparticles have emerged as a promising delivery system for telomerase therapies. LNPs can encapsulate mRNA encoding TERT and ferry it into target cells, leveraging the same technology proven by mRNA vaccines. This non-viral, non-integrating approach can transiently express telomerase in cells, avoiding the risks of DNA integration associated with viral vectors. Crucially, TERT mRNA delivered via LNP produces only temporary telomerase activity, after which the mRNA degrades and telomeres resume normal shortening. This transient effect is designed to reduce long-term risks like uncontrolled cell growth, while still achieving meaningful telomere extension. Preclinical studies in human cells and tissues. Early proof-of-concept studies demonstrated that introducing modified TERT mRNA into human cells can extend telomeres and rejuvenate cellular function. Human fibroblasts and muscle cells. In a landmark 2015 study from Stanford University, Ramunis et al. Showed that delivering modified TERT mRNA to middle-aged human fibroblasts and myoblasts increased telomerase activity for 1-2 days and extended telomere length by up to approximately 0.9%. Kilobases. With 3 serial mRNA transfections over 4 days, cells gained additional replicative capacity. Fibroblasts achieved approximately 2008 extra population doublings, effectively dramatically increasing their proliferative lifespan. Importantly, treated cells eventually did stop dividing and showed normal senescence markers, indicating they were not made immortal. This was a key safety finding. Telomerase rejuvenation can boost cell growth without permanently transforming cells. The researchers noted this rapid telomere extension method could aid tissue engineering and regenerative medicine. Progeria cells, telomerase mRNA has shown striking reversal of cellular aging in Hutchinson-Gilford progeria syndrome, a disease of accelerated aging. A Houston Methodist team led by Dr. John Cook transfected HGPS patient fibroblasts with ET-TERT mRNA, which dramatically restored HGPS cell proliferation and reversed markers of senescence. Treated progeria cells began to behave like healthy young cells, with normalized gene expression and growth rates. Essentially, transient telomerase expression reversed many features of cellular aging in this model of rapid telomere attrition. This proof of concept that mRNA can reverse cellular aging in human cells has spurred further collaborations in regenerative medicine. Human skin cells and tissue. Recent research has applied telomerase mRNA LNPs to human skin cells to improve regeneration. In 2024, Chang et al. Formulated H-TERT mRNA-loaded lipid nanoparticles and tested them on human skin cell suspensions used for grafting wounds. The optimized LNPs achieved efficient delivery and expression of telomerase mRNA in these skin cells. As a result, telomerase activity in the cells increased significantly, and markers of senescence and DNA damage were reduced. In a laboratory model of a skin graft, telomerase LNP treatment enhanced the engraftment and proliferation of human skin cells in a wound, leading to better regeneration of a skin-equivalent tissue in mice. In other words, telomerase mRNA in a lipid nanoparticle counteracts telomere shortening to improve wound healing. This finding suggests telomerase therapy could boost healing in aged or chronic wounds where cell senescence impairs recovery. These cellular and ex vivo tissue studies collectively show that telomerase mRNA delivered by LNP or similar methods can temporarily rejuvenate human cells, extending their telomeres and functional lifespan without permanent genomic changes. The success in human cells and 3D tissue models has paved the way for in vivo animal studies. Efficacy in animal models. Animal studies are validating telomerase LNP therapy in living organisms, including normal aging and disease models. Lifespan extension in mice. The concept of telomerase therapy increasing longevity was first proven using viral vectors. A pioneering 2012 study by Maria Blasco's group treated adult and old mice with an AAV carrying the TERT gene. The single treatment had remarkable beneficial effects, improving health metrics and significantly extending median lifespan by 24% in 1-year-old mice and 13% in 2-year-old mice. Notably, there was no increase in cancer incidence in telomerase-treated mice compared to controls, alleviating a major safety concern. This viral gene therapy study was a milestone, demonstrating that telomerase activation can delay aging and extend lifespan in a normal mammalian organism. It provided a strong rationale for pursuing telomerase therapeutics, though the field has since shifted toward transient mRNA LNP approaches for better control. Progeria mouse model. Building on cell studies in progeria, researchers applied telomerase therapy in a mouse model of HGPS. In 2021, Cook's team reported that telomerase mRNA therapy reversed vascular cell senescence and significantly extended the lifespan of progeria mice. Treated progeroid mice showed improved cardiovascular health and lived longer than untreated ones, indicating the therapy can ameliorate accelerated aging in vivo. This provides further evidence that transient telomerase delivery can systemically rejuvenate tissues and improve survival in an aging-related disease model. Lung fibrosis model. Short telomeres contribute to idiopathic pulmonary fibrosis, an age-associated fatal lung disease. A biotechnology startup, Rejuvenation Technologies Inc., has developed a lung-targeted TERT mRNA LNP and tested it in mouse models of IPF. According to their SBIR grant data, a single intravenous dose of TERT mRNA LNP in mice extended telomeres in vivo by an average of approximately 230 base pairs, effectively reversing the equivalent of years of telomere shortening in humans. In a telomere-deficient IPF mouse model, this treatment increased survival by 210%, reduced lung fibrosis by approximately 68%, and improved lung function by approximately 58% compared to controls. These dramatic improvements suggest telomerase LNP therapy not only lengthened telomeres, but also helped regenerate lung tissue and counteract disease processes. Importantly, the telomerase activity was transient, after which normal telomere shortening resumed, preserving the natural tumor suppressive mechanisms. Wound healing in mice. The aforementioned 2024 wound healing study also included an in vivo component. Researchers created partial thickness wounds on mice and applied human skin cell grafts that had been treated with HTERT mRNA LNP. The telomerase-boosted skin grafts showed superior engraftment and healing. More human cells survived and proliferated in the mouse wounds, and there were fewer senescent cells in the regenerating tissue. This in vivo validation supports the idea that telomerase therapy could improve outcomes in wound treatments, especially in aged patients. Across these models, telomerase delivery consistently exhibits therapeutic effects related to aging, improving tissue repair, organ function, or survival. The transient mRNA LNP method in particular shows promise for safe, repeatable dosing. It's worth noting that while permanent overexpression of telomerase in mice can cause cancer, using controlled, episodic telomerase activation may avoid that risk. The animal data so far indicate a favorable balance, rejuvenation benefits without overt oncogenic effects. Given the encouraging preclinical results, researchers and companies are now progressing telomerase LNP therapies toward clinical application. Here is the current state of clinical trials and human-focused research. Pulmonary Fibrosis, Rejuvenation Technologies Inc. is advancing a telomerase mRNA LNP therapy for pulmonary fibrosis toward human trials. Their lead candidate is an optimized TERT mRNA in a custom lung-targeted LNP, aiming to restore telomere length in lung cells. RTI has received substantial funding to complete IND-enabling studies. These studies include pharmacokinetics, dose finding, safety, toxicology, and comparisons to existing IPF drugs. The goal is to submit an investigational new drug application to the FDA, potentially leading to a Phase 1 clinical trial in IPF patients. If successful, this would be one of the first human trials of telomerase via LNP. As of late 2023, RTI announced $10.6 million seed financing to ramp up manufacturing and clinical operations, explicitly to advance its programs to IND approval in fibrotic diseases. This suggests a Phase 1 trial in these indications could begin in the near future. Skin rejuvenation and wound healing. A collaboration between Houston Methodist Hospital and Avita Medical is translating the telomerase mRNA approach into a therapy for skin repair in burn patients and potentially skin rejuvenation for anti-aging. Avita's proprietary spray-on-skin system, which is an FDA-approved method to apply a suspension of a patient's skin cells to wounds, is being combined with Houston Methodist's telomerase mRNA technology. In this approach, cells harvested from an elderly patient's skin would be treated ex vivo with TERT mRNA LNP just before being sprayed onto the wound in order to improve graft take and healing. This program is currently preclinical. As of 2020, the collaboration established proof of concept, and Avita secured an exclusive option to license the telomerase technology for skin applications. The initial studies aim to generate data for a regulatory submission. Avita has indicated a plan to pursue an IND for this mRNA-based skin rejuvenation therapy, which could lead to clinical trials in burns or possibly aesthetic dermatology. No human trials have begun yet, but the partnership signals a clear commercialization intent once safety and manufacturing are worked out. First-in-human telomerase gene therapy, although not using LNPs, it's relevant that Libella Gene Therapeutics launched the world's first clinical trial of a telomerase therapy in humans, aiming at biological aging itself. In late 2019, Libella announced an IRB-approved Phase 1 trial in Colombia to test an AAV-based HTERT gene therapy in volunteer subjects, with the goal to treat and reverse aging. Participants receive an infusion of an adeno-associated virus carrying the telomerase gene, intended to lengthen telomeres in their cells. Libella's trial is noteworthy as a pay-to-play anti-aging trial operating outside the U.S., with each volunteer paying $1 million for the experimental therapy. The company's chief scientist, Bill Andrews, stated that HEART is delivered via AAV and that this approach appeared safe in prior studies. While this is not an LNP method, it marks a translational milestone, demonstrating the demand and willingness to test telomerase therapy in humans for age reversal. However, being an unorthodox trial, it has been met with skepticism. As of now, no results from the Libella trial have been published, so its outcomes and current status remain unclear. It does highlight that telomerase-based interventions are entering human testing, albeit through alternative pathways. Other prospective clinical uses, researchers are also eyeing telomerase mRNA for specific age-related disorders or telomere syndromes. For example, there is interest in applying TERT mRNA therapy to diseases like idiopathic pulmonary fibrosis, liver cirrhosis, bone marrow failure, and even immunosenescence. Rejuvenation Technologies has mentioned early-stage work on targeting the aging immune system with telomerase mRNA. Another area could be cardiovascular aging. Telomere shortening in heart cells contributes to heart failure, so telomerase therapy might one day be tested for cardiac rejuvenation. Indeed, in mice with heart disease, telomerase gene therapy improved cardiac function. As of 2025, no formal clinical trials in these areas have been registered, but these are logical future directions as more safety data emerges. It's expected that initial human trials will focus on serious diseases rather than healthy aging to satisfy regulatory requirements, but positive results could pave the way to broader anti-aging applications. In summary, no major clinical trial of telomerase LNP in humans has been completed yet, but multiple paths are converging toward first-in-human studies. The most concrete near-term trial will likely be RTI's fibrotic disease program, followed by the Avita skin rejuvenation trial. On the fringes, Libella's ongoing experiment targets biological age. Meanwhile, any approved use of telomerase therapy will mark a breakthrough in gerotherapeutics, as regulators have never before evaluated a treatment explicitly aiming to reverse cellular aging. Companies developing telomerase LNP therapies. A number of biotech companies and research institutions are actively working on telomerase lipid nanoparticle therapies, reflecting growing commercial interest in anti-aging and regenerative medicine. Key players include Rejuvenation Technologies, Inc., a Silicon Valley startup focused on mRNA therapeutics to address mechanisms of aging, prominently telomere shortening. Co-founded by Dr. John Ramunas and Dr. Glenn Markoff, alumni of Helen Blau's Stanford lab, the company has invented a tissue-targeted TERT mRNA LNP platform that can rewind years of telomere shortening with a single dose. RTI's pipeline targets age-related diseases. Their lead programs are for pulmonary fibrosis and liver cirrhosis, and they are exploring immune system rejuvenation. They have raised significant funding to move toward clinical trials. RTI's technology is partly based on the seminal Stanford work and subsequent proprietary LNP innovations. They likely hold or license key patents on telomerase mRNA therapy, with a team that now includes a chief medical officer specializing in lung disease. RTI is a frontrunner in bringing telomerase LNP therapy to the clinic. Chromex Biodevelopment, a company co-founded by Dr. John P. Cook of Houston Methodist, aiming to develop telomerase mRNA therapies to treat cellular senescence. Cook is an inventor on patents for using mRNA telomerase against aging, and his lab's research forms the basis of Chromex's technology. While details on Chromex Bio are limited publicly, it appears to be focusing on therapeutic applications of telomerase mRNA, potentially in areas like cardiovascular aging or wound repair. It may be in early development stages, collaborating with Houston Methodist Center for RNA Therapeutics. Cook's conflict of interest statements also note sponsored research with VGXI and Avita Medical, indicating Chromex Bio might be aligned with those partners for manufacturing and skin applications respectively. In summary, Chromex Bio is another startup in this niche, translating academic research on telomerase LNP into therapeutics. Avita Therapeutics, this established regenerative medicine company, is not developing telomerase LNPs on its own, but through the collaboration with Houston Methodist, it has an exclusive option to commercialize telomerase mRNA technology for skin rejuvenation. Avita's interest is to enhance its skin graft product for use in older patients, burns, chronic wounds, and even cosmetic dermatology. The joint project is currently preclinical, but Avita's involvement is significant because it brings an existing commercial infrastructure and regulatory experience to the table. If the telomerase rejuvenated skin graft approach proves viable, Avita could fast-track it into clinical trials and eventually incorporate it into their product line for surgeons. Essentially, Avita could become the first to market a telomerase-augmented cell therapy, specifically for dermatological use, if all goes well. They have publicly acknowledged this pipeline expansion into modified cells delivery for reversing skin cell aging. Libella Gene Therapeutics, a Kansas-based company taking a gene therapy approach to telomerase. Libella's method uses AAV vectors rather than LNPs, but they target the same outcome telomerase expression to. Lengthen telomeres. Libella made headlines by seeking the world's first anti-aging therapy, offering its heart gene therapy in a self-funded trial in Colombia. They claim to have a proprietary AAV delivery system for telomerase that has shown safety in other trials. The company's focus is explicitly on aging as an indication. While Libella's approach is outside mainstream regulatory channels, it underscores the commercial interest in telomerase therapies. Libella has likely secured IP around AV delivery. However, their progress and credibility are often debated in the gerontology community. They are not selling a product on the open market, but effectively selling access to experimental therapy. Libella's endeavor, along with BioViva's, can be seen as a high-risk early commercialization attempt in the anti-aging telomerase space. BioViva Sciences, another company that garnered attention by pursuing telomerase gene therapy. In 2015, BioViva's CEO Elizabeth Parrish reportedly underwent an experimental telomerase gene therapy as a personal trial. BioViva claimed some telomere lengthening in her white blood cells afterwards, but no peer-reviewed data were published. BioViva has since partnered with research groups to offer gene therapies via clinics abroad. Like Libella, their methods involve viral vectors and are outside regulated clinical trials. While BioViva is not working with lipid nanoparticles, it is notable historically as one of the first private entities to attempt telomerase therapy in a human. They highlight the demand for anti-aging interventions, though their approach has been criticized for lack of oversight. BioViva does not have an approved product and operates more as a venture-backed translational outlet for experimental gene therapies. In addition to these, large pharmaceutical companies have not yet entered the telomerase LNP arena in a visible way. However, the rapid advancement of mRNA therapeutics means bigger firms could eventually explore telomerase as a target for regenerative medicine. For now, the field is dominated by startups and spin-offs from academia, holding the key IP and know-how. The telomerase mRNA therapy concept is protected by a family of patents arising from the Stanford Houston Methodist Research. Key inventors filed broad patent applications around 2013-2014 for compounds, compositions, methods, and kits relating to telomere extension. These have since been granted as U.S. patents. For example, U.S. Patent 10,525,075 and U.S. 11,007,210 cover the transient delivery of modified TERT mRNA to elongate telomeres. The patents likely claim the use of nucleoside-modified mRNA encoding telomerase for therapeutic telomere extension, as well as specific formulations and dosing regimens. Stanford University and Houston Methodist jointly own some of this IP, and their licensees include the companies mentioned. Beyond that core IP, companies are filing follow-on patents for specific applications or improved delivery systems. For instance, Rejuvenation Technologies may have proprietary LNP formulations optimized for lung delivery. Any unique targeting ligands or ionizable lipids they developed could be patented. Likewise, methods to rejuvenate skin grafts with mRNA could be subject to new patents under Avita's agreement. In short, a nascent patent estate is forming around telomerase reactivation therapies, which will be crucial for commercialization. As of now, no patents specifically on small molecule telomerase activators for anti-aging have proven clinically relevant. The gene mRNA therapy route is the primary focus of IP. Safety and regulatory considerations. From a regulatory standpoint, telomerase LNP therapy is a novel category. Key considerations include safety. Because telomerase can enable cells to bypass senescence, a major concern is the potential to promote cancer. Regulators will scrutinize animal data for any sign of tumorigenesis. The reassuring news is that transient mRNA delivery appears to avoid this pitfall. In cell and mouse studies, telomerase activity spikes for only approximately 24-48 hours, then shuts off. This transient expression means treated cells still eventually senesce. Also, experiments in mice showed no increase in cancer over the animals' lifetimes when telomerase was activated in adults. Nonetheless, human trials will likely start with severe diseases and careful monitoring. Regulators may require extensive toxicology studies before allowing trials in conditions that aren't immediately life-threatening. Efficacy endpoints. demonstrating tangible benefits in humans will be critical. In diseases like IPF, endpoints could include improved lung function or fibrosis on imaging. For a wound healing indication, endpoints might be faster closure or better graft survival. Because aging itself is not an FDA-recognized indication, companies target specific age-related diseases or conditions. However, a successful trial in one area could encourage off-label or future trials in broader geriatric use. Regulatory agencies may also consider biomarker endpoints to support the mechanism of action. Manufacturing and quality. mRNA LNP products must meet stringent manufacturing standards similar to other nucleic acid therapies. Companies are scaling up production and working on cold chain storage, particle uniformity, etc. The involvement of experienced manufacturers is aiding this process. Any clinical-grade telomerase mRNA will use pseudouridine and 5-methylcytidine modifications to reduce innate immune activation, and LNP formulations likely draw on known lipids. Patents show the formulations tested include common ionizable lipids and pegylated lipids, giving regulators familiarity from prior LNP drugs. Ethical considerations, especially for anti-aging uses. Ethical questions arise. Telomerase therapy in otherwise healthy older adults would be a bold step. Regulators will likely demand substantial evidence of safety and benefit in age-related disease before countenancing enhancement uses. The Libella trial's pay-to-participate model has been controversial, highlighting the tension between patient autonomy and regulatory protection. As this field moves forward, expect discussions about how to safely test therapies that blur the line between treating disease and enhancing longevity. Telomerase LNP technology is at the cutting edge of therapeutic development, and its future directions are poised to transform aspects of medicine and aging research. Treatment of telomere biology disorders. Beyond normal aging, there are hereditary telomere shortening disorders. These conditions could be prime candidates for telomerase mRNA therapy. For example, hematologists might use telomerase LNP infusions to restore bone marrow function in patients with telomerase mutations, as an alternative to bone marrow transplant. Early supportive data comes from experiments where transient telomerase expression rescued cells from telomere-driven crises. We may see compassionate use cases or small trials in these rare diseases once initial safety is shown. Regenerative medicine adjunct. One immediate practical use of telomerase LNPs is as an adjunct to cell therapies. The Stanford team originally noted it could help in tissue engineering. This is now happening with the skin graft example, essentially juicing up a patient's own cells to proliferate better and heal wounds. In the future, similar approaches could be used for cell transplants in other contexts, e.g., rejuvenating T cells or stem cells before infusion to improve immunotherapies, or treating donor organs ex vivo to refresh cell populations before transplantation. Telomerase mRNA could be applied transiently to expand cells for therapy. These applications might progress faster because they modify cells outside the body and then use them in established transplant procedures. Combining with other anti-aging strategies, telomerase activation addresses one hallmark of aging. Future anti-aging regimens might combine telomerase LNP therapy with other interventions like senolytics, parabiosis mimetics, or stem cell therapies. By tackling multiple aging mechanisms, the hope is for synergistic effects on health span. For instance, periodic telomerase LNP treatment might keep tissue's telomere reserves sufficient, while senolytics clear out any cells that still become senescent, a 1-2 punch for tissue rejuvenation. Research into dosing frequency will be important. How often would an older adult need telomerase mRNA treatment to maintain cellular function? Animal studies suggest benefits from even a single dose lasting for months, but this will need exploration. Broader geroprotection. If telomerase LNP therapy proves safe and effective in specific diseases, it could herald a new class of geroprotective therapies. Eventually, clinicians might use telomerase LNP infusions prophylactically in high-risk aging populations. For example, giving an elderly person a telomerase boost prior to a major surgery to aid recovery, or treating frail individuals to improve their resilience. While this is far off and would require extensive validation, it's within the realm of possibility if incremental steps succeed. Indeed, the long-term vision shared by some of the scientists is to employ RNA therapy to improve health span in the elderly. Telomerase is one of the first such interventions moving from theory to potential practice. In conclusion, telomerase mRNA delivered via lipid nanoparticles represents a cutting-edge convergence of gene therapy and geroscience. The current state of research shows compelling rejuvenation effects in cells and animals, from extended cell lifespans, to improved tissue repair, to prolonged life in mice. Clinical translation is underway. Companies like RTI and collaborations like Houston Methodist Avita are steering this technology toward human trials in the next few years, targeting conditions of aging and degeneration. While no human has yet received a telomerase LNP therapy in a formal trial, the groundwork has been laid. If these therapies succeed, they could inaugurate a new paradigm of rejuvenation medicine, where aging at the cellular level can be reversed to treat disease and possibly slow the aging process itself. Of course, challenges remain. Safety must be rigorously proven and public acceptance earned, but the progress to date is highly encouraging. The coming decade will likely reveal whether telomerase therapies can fulfill their anti-aging promise in humans, turning laboratory breakthroughs into real-world longevity interventions.